In this video, we're going to talk about culturing microorganisms, which is a biology-only topic. When conditions, such as temperature and availability of nutrients, are suitable, bacteria will divide by a process called binary fission. This is where the cell makes copies of its genetic material, its DNA and plasmids, before dividing into two daughter cells. Some bacteria will be able to divide as often as once every 20 minutes, meaning one single bacterium can divide to form a colony of millions of bacteria in a very short period of time. This increase is called exponential growth. Bacteria can be grown or cultured in or on a culture medium, which is either a liquid nutrient broth or an agar gel. It's important to ensure that the culture isn't contaminated by other microorganisms, so sterile or aseptic conditions are used. Firstly, all of the equipment is sterilised using an autoclave, in which they are heated under very high pressures. This makes sure that there are no microorganisms already present on the equipment before starting the experiment. Then, the inoculating loop, which is used to transfer microorganisms to the culture medium, is sterilised. This is done by passing the loop through a Bunsen burner flame until it burns red hot. This is done before and after use, to prevent contaminating the culture medium and also to prevent contaminating the environment. Finally, if you're culturing bacteria using agar gel, the lid of the Petri dish containing the culture is secured to prevent bacteria from contaminating the environment. The lid should be secured using two pieces of tape, but should not be sealed completely, as this would produce anaerobic conditions, which are conditions lacking oxygen. This would promote the growth of pathogens, which are bacteria dangerous to our health. The Petri dish will then be placed upside down and cultured at 25 degrees for 20 to 48 hours. Bacteria would produce more quickly at higher temperatures, but incubating cultures at temperatures closer to the human body temperature, which is 37 degrees, would also promote the growth of pathogens. That's why in school laboratories, the maximum temperature for culturing microorganisms is around 25 degrees. Once the experiment is complete, Petri dishes are autoclaved before being disposed of, again to prevent contamination of the environment. Your second required practical activity is found in this topic. Antibiotics and antiseptics prevent bacteria from growing or reproducing. You can investigate the effect of different antiseptics on the growth of bacteria. To do this, you will culture bacteria on an agar plate, ensuring all aseptic conditions are followed. The agar plate will then be divided into 3 equal sections, and a paper disc soaked in different antiseptics will be placed in the middle of each section. The plate will then be incubated for 48 hours. After incubation, a clear zone, known as a zone of inhibition, should be visible around each disc in each section. Measure the diameter of each clear zone. The antiseptic which produces the zone of inhibition with the largest diameter is most effective at treating the bacteria used in the experiment. You can also calculate the area of the zones of inhibition and use this to work out which antiseptic is most effective. To do this, measure the radius of the clear zone, which is half of the diameter, and use the equation area equals pi r squared. If you know the mean division time of a bacterium, you can also calculate the number of bacteria in a population after a certain time. For example, if a bacterium has a mean division time of 20 minutes, let's work out the population size after 2 hours. 2 hours equals 120 minutes. 120 minutes divided by 20 is 6. Therefore, the bacterium will divide 6 times in 2 hours. To work out the population size after this time, calculate 2 to the power of the number of divisions. 2 to the power of 6 is 64. Therefore, after 2 hours, the number of bacteria will be 64. Higher tier students should also be able to express the number of bacteria in standard form. Let's look at another example. If the bacterium, which has a mean division time of 20 minutes, is allowed to divide over a period of 6 hours, 18 divisions will occur. 2 to the power of 18 equals 262,144 bacteria. In standard form, this can be written as 2.62 times 10 to the power of 5.